For many centuries, the foundational history of ancient Egypt was constructed entirely on top of a biological myth. The mid-20th century historiographical baseline in European Egyptology actively severed ancient Egypt from the rest of the African continent. The academic establishment attributed the origins of Egyptian civilization exclusively to Eurasian or Mediterranean populations. They justified this consensus using culturally biased philology, the study of language in historical texts, and subjective interpretations of art, rather than relying on hard biological science. The philosophical underpinning of this era leaned heavily on European thinkers like Hegel, who asserted that Africa possessed no historical past of its own. Defeating an entrenched historical myth of this scale required abandoning traditional historical methods entirely. The problem had to be attacked through physical chemistry. Dr. Cheikh Anta Diop entered the debate as a rigorously trained scientist. He held an elite academic pedigree, studying physics and chemistry in Paris under the Nobel laureate Frédéric Joliot-Curie. Diop deployed a transdisciplinary approach. He merged physical chemistry, paleontology, and anthropology to create an entirely new historiographical protocol. In Dakar, he established the very first radiocarbon dating laboratory on the continent of Africa. Radiocarbon dating is a technique that measures the decay of radioactive carbon-14 to precisely determine the age of organic materials, generating objective, empirical data. Diop's core thesis was specific. Ancient Egypt was a Black African civilization, and he intended to prove it using immutable biological markers. He recognized that rhetoric could never defeat colonial assumptions. He needed undeniable microscopic proof to force a shift in the academic consensus. The 1974 UNESCO Symposium in Cairo served as the formal arena for an international debate on the peopling of ancient Egypt. The academic establishment relied heavily on a primary shield, the argument that embalming materials obscure a mummy's original skin tone. Chemicals like natron, a natural salt used to dry tissue, and embalming resins heavily degrade and darken the upper layers of mummified skin over millennia. To counter this, Diop developed the melanin dosage test. This was a specialized biological analysis designed to calculate the original pigmentation of ancient populations through their physical remains. Diop acquired highly restricted mummified skin samples from the Mariette excavations housed in Paris to conduct his tests. This microscopic cross-section of ancient skin illustrates the mechanics of Diop's test. He bypassed the destroyed upper epidermis, extracting tissue strictly from the boundary between the dermis and epidermis. The chemical reality Diop exploited is that melanin is largely insoluble. It survives intact for millennia, despite heavy external chemical additives. Observing the tissue under ultraviolet light, Diop revealed the melanin concentration perfectly aligned with the high density of sub-Saharan African populations and was non-existent in leucoderm ranges. At the Cairo Symposium, Diop physically placed his microscope slides on the table and invited opposing scholars to look through the lens themselves. By presenting the melanin dosage test, Diop successfully shifted the debate away from subjective cultural interpretation and grounded it strictly in immutable physical chemistry. The second pillar of empirical data Diop presented at UNESCO was based in physical anthropology and osteology, the scientific study of bones. Mid-century Egyptologists preferred a tool called craniometry, which is the measurement of the skull.
The specific flaw of craniometry was its selective focus. Researchers relied on long facial indices to categorize skulls as Eurasian, while ignoring the post-cranial measurements of the rest of the body. Diop systematically rejected craniometry entirely, labeling it an arbitrary and flawed metric that was easily manipulated by researchers to fit pre-existing colonial assumptions. He introduced an empirical alternative, the Lepsius canon, a mathematical standard established by a 19th-century German scholar. This 3D wireframe skeletal projection demonstrates the Lepsius canon. It demands measuring post-cranial proportions, forcing an analysis of the entire skeleton from the neck down. Diop focused heavily on the specific metrics of the canon, noting the ratio of arm length relative to torso length, applied to the perfect Egyptian anatomical standard. The osteological conclusion was exact. The post-cranial proportions of the ancient Egyptian strictly align with the negroid physiological archetype, featuring characteristically shorter arms. By proving that analyzing the whole body eliminated the ambiguity constructed by earlier researchers, Diop turned bone structure into undeniable demographic data. The third dataset presented at the 1974 symposium was serology, the study of blood serum and its properties. Blood groups map ancient population distributions exactly as they existed before modern genetic admixture. Diop established the geographic baseline for blood group A2 in Antiquity. Before extensive intermingling, the A2 blood group was strictly characteristic of Eurasian and white populations. He contrasted this by explaining the geographic baseline for blood group B. This map displays the baseline distribution of these blood groups. It correlates blood group B with high-density clusters found among West African populations, separate from the A2 clusters in Europe. Diop then introduced the serological data extracted directly from ancient Egyptian mummified tissue. Tissue from those mummies, particularly from Upper Egypt, clustered heavily in blood group B. Diop used this specific data point to challenge the room, explicitly ruling out a Eurasian biological origin for the Nile Valley populations. Through blood typing, Diop had mapped an invisible internal biological lineage that connected the Nile Valley directly to the heart of the African continent. At the climax of the 1974 Cairo Symposium, Diop and his colleague Théophile Obenga had cornered the establishment. The official conclusion of the UNESCO International Committee stated that Diop's presentation was painstakingly researched, while the opposing scholars were utterly unprepared to debate on empirical grounds. 50 years later, the modern era of archaeogenetics has closed the gap between Diop's physical chemistry and current science. In 2025, the journal Nature published a breakthrough study detailing the first complete whole genome sequence of an Old Kingdom Egyptian. The predictive DNA analysis confirmed dark skin pigmentation and an overwhelming majority of African ancestry. Concurrently, the Agenda project, assessing genetic diversity in Africa, is adding thousands of new whole genome sequences to global databases, furthering Diop's goal of mapping human origins through African biological diversity. Modern DNA sequencing has ultimately operated as the final validation of Diop's melanin dosage test and his transdisciplinary methodology. You cannot rewrite history with mere philosophy. To reclaim the truth, you must arm yourself